legacy is a tremendously important thing, and much of what our lovely Dr. Dennis Tarnow touched on there is not only about what you contribute in this world, but it's about the legacy you leave when you pass to the next. And I know that for those people who've experienced the loss of a parent, legacy is very important. How much more incredible must that be when your father has literally touched the whole of humanity through his works and his discovery? How proud it must be for our first speaker to carry that surname which is most famous in this great academy. I'm very privileged to introduce to you our keynote speaker for today, Rickard Brånemark, who is a research scientist who has dedicated his career not in dentistry, but in the field of orthopedics, as associate professor in Gothenburg and professor emeritus at the University of California here in San Francisco. But he's now CEO and founder of Integrum. This is a company that helps to make the lives for amputees profoundly better through the miracle of osseointegration, but also through the integration, if I can use that word, and application of neurophysiological communications, which I guess he will touch on. So it's a great honor for me to welcome to this podium Dr. Rickard Brånemark.
Okay. Good afternoon. This is my first time at the Academy of Osseointegration, so I will try to be brief because I only have about 60 years of research to cover. And as you see here, I have many different hats, and we're trying to cover most of them. So I've mostly been working on amputees and extremity reconstructions. And here you see the famous reconstruction of Darth Vader from Star Wars. So to try to do something that's never been done before, maybe it's in the family, but you also need to have a certain mindset. You might need to be stubborn, but you also need to look into the world from different perspectives or different angles to see how you can fit new things together in a way that hasn't been fit together previously, but maybe the bits and pieces of the puzzle been there already. So, you know, this is the beginning of osseointegration. I guess you know about this implant, the titanium implant. But did you know where the titanium came from? Well, I'm an orthopedic surgeon. I'm happy to say that it came from an orthopedic surgeon that PI talked to many years ago and asked for a good material to use for his vital microscopy chamber. And maybe you also know that PI had a bit of a temper. So when he tried to remove this experimental implant, he couldn't get it out. And because of its temper, it got sort of destroyed. And that was just a pity for him in the early days when the funding was not so good as it became later. Do you know what this is? This is an arm of a retired professor of hand surgery that was a PhD student in my father's lab in the early 70s. because he didn't just look into a microcirculation in bone tissue, he also looked into microcirculation in living humans. And here he's looking into what was the first time it could be visualized. So these are red blood cells in man in the capillary. And if you were a PhD student in the lab in those days before, IRBs and stuff like that, there were a few things you had to go through. And maybe you know that the work continued also with other types of microcirculation and healing of bone defects. And maybe you've seen this before. Maybe you've seen this before. This is one of the first osseointegration cases ever in the world. One of the beagle dogs. Maybe that was our watchdog when I was a kid. I don't remember. So anyway, osseointegration, what is it really? Well, frankly, we don't know. So this is one of the definitions. And if we try to interpret what it really is, in principle, it is a way to transfer loads from a biomaterial to bone tissue without creating inflammatory reactions in the interface. And that's why we can maintain the interface. If we get this fibrous tissue interface that you used to get for orthopedic implants in the 60s, you get a movement of the biomaterial towards the bone tissue that's creating inflammation, further fibrous tissue, and then finally the implant will fail. And we all know that it took about 20 years to convince the clinical society in dentistry, and then it's nicely talked about orthopedics also. That took a long time to convince them that this is something that is actually possible. So this is one of my father's patients, and this is one of his old slides. 1999, it was a 30-year follow-up. I used this picture to illustrate that, okay, titanium is a fantastic material, but which is really the best material in the world? Bone tissue, of course. So it's fantastic that bone tissue can tolerate a couple of tiny implants for many, many years without reacting it. So one thing is dentistry, then moving into permanently skin-penetrating implants that started in 1977, that is leading towards what I'm dealing with now, amputation care. That was also fairly controversial, because they say, you have dentition already, you have the mucosa, it's very good, vascularity, etc. But permanently skin-penetrating devices, they will never work. But we know that they can work in good, healthy tissue condition, and also in the few very compromised patients. And this is patient treated by my father also with post-op radiation therapy, damaged soft tissue, damaged bone tissue, and still with a great team effort with dental technicians creating a new face and a new environment and a new life, which is what we're here for. So because something is rigidly connected to the bone, you can actually use that to hear. And we can even show that amputees can hear with their implants. And this is just by transferring sound here for the bone-anchored hearing aid directly to the inner ear. So what can we do? Can we move this further? So in the early 2000s, we started to think about what can we do. Can we make this better, safer, faster, stronger? Can we do something better than the original machine surface? That was what I had been taught all the time, that is the best. What can outcompete 50 years of clinical experience? Can faster also be better and safer, which is like a contradiction? And how much stronger can we make osseointegration? Do we really know everything about the interface and the composition, how to work with that? So if we do change something that is working fairly well, what are really the risks? What if we set up more aggressive treatment protocols? If we go outside the established indications to new unproven indications, will that increase the risk for loosening, and what about the peri-implantitis? So we've been working with something that we call the Bio-Helix concept. It started about 2002. I mostly worked with Peter Thompson. And if you don't know, Peter Thompson is now a retired professor, and he took the chair after my father. So when we started off, we had started to see that a new surface is coming up, they're rough, and there seems to be a bit better, but the machine surface has a lower frequency of peri-implantitis. How can we fit this together? And then you start to look into biology, which is utterly complex. Totally impossible to really understand how things are working there. But we said, okay, let's focus on the osteoblast. So how can we predict that the osteoblast will produce bone around an implant? Well, we need to dig into what's really happening inside the cell and how the cell is communicating with the surface. We need to go down to the cell membrane level, and we would like to see what's happening there. And to be able to do that, we used a new technology in those days, originally developed for microelectronics, called focused ion beam. And if you have a soft tissue and a hard material, it's very difficult to look into that specific interface with high resolution. But with the focused ion beam technology, you can do that. You put a thin piece of platinum to keep the two different materials together. You do this in an electron microscope. You grind down around the platinum, and then you can lift up a very, very thin slice of the material. You can put it, and then with soft ion beam technology, trim it down so it's so thin, so it's about 1,000 of a human hair. You take a human hair and you divide it in the longitudinal direction 1,000 times. That's how thin it is. And when it is so thin, it becomes transparent in high-resolution transmission electron microscopes. So you can start to see a picture, a high-resolution picture of the interface. And we developed a laser micromachining setup to be able to create a surface that could enhance osseointegration. And we positioned it only in the valleys and not close to the marginal bone, so only about 40% of the surface. So if we now go into micro and nanocosmos, you see here, this is finally the structure that the cell membrane will see. And these spheres are about 1/10 of a human red blood cell. And you see on top of them, there is a lot of small irregularities. And it's actually these irregularities that sort of communicate with the cell membrane. And here is a high-resolution picture where we can see a thin layer of titanium oxide. And here we can also identify the structure of the oxide. This is a rutile phase that at least some believe is a better oxide than the anatase. But I think this is the most interesting picture. Oh, let's see if I can use this. Okay. So here you see, this is titanium, and you see titanium here. This is the oxide layer, which is here, and this is bone tissue. This is the scale, 200 nanometers. Very, very small dimension. And what we can see here is that it's not so that the bone is in close contact. The red here is calcium. You have calcium inside this nanoporous oxide. So that is the bone is inside the implant. Okay, so what will that mean? Well, by just modifying 40% of the surface, we can, by using this technology, kick up the biomechanical capacity about 250%. And we can show that it's the bone tissue surrounding the implant that is the weakest part. The interface is so strong, so the fracture will be out here in the bone tissue. So what is this? Well, here we show some pictures, okay, mostly mechanical pictures, but isn't it the most important that the bone cells really like the surface? So this is a technology that is removing all the extracellular matrix. So here you see the cell, and you see how the cell is actually really grasping the surface. And this is an indication that the cells like the surface. So we've done tons of scientific evaluations on this. We studied cultured, we studied it animal studies, and see here we worked with titanium grade 1, short-term studies, long-term studies, and also some clinical studies. This is on dental implants, 5-year follow-up. And here's some scientific references. And of course, this is for me also work to pave into more demanding mechanical applications for orthopedics. So what is this? Okay, this is the only case in the world. Who's gonna hit it? I don't think you will hit it. This is a bearded vulture. So this is a species that is almost extinct in Europe, and they're trying to rebreed it. It's a female, she got an amputation, and she was about to die because they can't feed themselves if they don't have 2 feet. So this is, to my knowledge, the only osseointegrated bird ever. A little bit maybe off topic, but anyway. So let's move into orthopedics. I know that Jonathan Forsberg was here maybe last year and talked about this, so I tried to be a bit brief. So what is this? This is a patient with an above-knee amputation and is using a socket. See it as a denture. The problem here, fundamentally, is a biomechanical problem. Because how can you transfer the loads from the socket into the bone through the soft tissues with skin and, you know, the thick thigh muscles without causing damage? You cannot, and the retention will not be good. That's why it's a lot of problems associated with socket prosthesis, as you can see here from a study we did a little bit more than 20 years ago, showing that there's a lot of problems just associated with the socket. So if we could get rid of the socket, it would be much better. And that is what we do with the osseointegration amputation prosthesis. And here you see that there is really nothing touching the soft tissues. All the loads go, like dental implants, directly into the skeleton, a much more favorable biomechanical situation. So this is the OPRA implant system that I've developed in Sweden now for many years. And in principle, I tell everyone, it's like a big dental implant. There's a fixture, a threaded fixture, and there is an abutment. So osseointegration in orthopedics, it's starting to be fairly well known and well understood. There is still a very, very big challenge to have something permanently penetrating the skin. So that's why we talk about the stage 2 surgery. So when you open up after implanting just the fixture, what we do is a total contradiction to what you typically do for amputation surgery, where you try to put all the muscles around and over the skeleton to don't have the skeleton sort of almost penetrating through the skin. Here we remove everything. We say we do a guillotine amputation of the muscles. We suture them into a muscle platform. You see that the bone is protruding out through that muscle platform. And then we take the skin and we even trim down the skin so it's super thin. It's like a full thickness skin graft, and then we put that directly on top of the bare bone and we suture it to keep it still. And here is a live picture. So you see, here's the muscle platform with the femur bone in the middle, and here is the trimmed down skin, and then you fold that directly on top of bare bleeding bone. It's very important that the bone is bleeding because the capillaries will sprout from the bone tissue and revascularize the skin, otherwise it will die. So here is directly post-operative, and these are pictures of the 5 years. So the trick here is to have this region not attached to the implant, but attached to the bone, and that we can do reliably. And if it's attached to the bone and there is no relative motion, no relative motion, no fibrous membrane, no granulation tissue formation, then biology will take care of the rest. The few bacteria that will come in there, the body will kick out. And we know that now, and our follow-up is 30 years. Okay, fine. So what does it really mean? A young Spanish boy, he was 19 years old when he had a terrible car crash and lost both legs. I saw him in Spain. He was sitting in a wheelchair. He was overweight. He was smoking. He didn't look up. His life was over. So we put, together with the Spanish team, we put implants in both of his legs. And here is in the beginning of his rehabilitation. He's now able to walk again. But look at his face. He's proud, he's the king, he's alive. That's what it's all about. Bringing people back to life. So let me talk about a few things that I think it's important for dentistry, but maybe especially also for orthopedics where the loads are a bit higher. So I like biomechanics, so let's talk about mechanics, axial rotation, bending, implant design, and kinetics. So how should we really think about this? Sometimes it's easy to do it wrong, because this is how we should think about it. And then you say, oh, what the heck is this biology? I don't understand a thing. I don't either, really, the details. But what we need to do is we need to work together with biology. You can't kill biology. You remember, why do we have traumatic surgery? Why do we take good care of bone tissue? Because if we kill it, it will not help us to get the result. That's why bone tissue is a fantastic tissue if you really take care of it. So this is an old picture. It's a bit blurry maybe, and that's not the more important. It's actually from Peter Thompson's work, studying the different phases, what's happening in the interface region. And what it's showing here, there is a lot of things happen early on, and then there's some things happening later on. And we need to pay attention to both those different phases. And why is that? Well, I hope you read my PhD thesis, because this is work from that. And this is showing that, and this is actually even before the Bio-Helix surface. So here it's showing, which I think is very interesting. So here is the starting point. So this is primary stability day 0 when you put in an implant. And then you see, okay, this goes up, good. So what is that? This is axial loading, because we have the screw shape, okay? That's transferring loads nicely. But what about the interface characteristics? You see, this is going down here, because we get some damage still in the interface region, and that needs to be remodeled, woven bone formation and locking in, and then over time it's starting to go up. And you see it gets substantially stronger over time than the primary stability at insertion, which is important to remember. Biology will help us to get a higher mechanical performance. So we can work with the implant design. Of course, for orthopedics, we have bigger implants. We can work with something called mechanical integration. How do we work with thin walls? What about undersize and compression? So there are a few different things to consider here. I don't think it's fully developed yet, and we're working a lot with it. So here, for instance, there is a way to, for an ordinary screw, it's just the first threads that will cover load. But by changing design, you can transfer load over a bigger region, which means that it's a more uniform stress distribution and should potentially be better for the bone. It's also so with the thin walls that certain designs will more, you see, it's almost like sort of fracturing the thin wall, which of course for a thin bone will not be able to walk on it. And here you see this is more of a preserving thing. And it's also so that with the designs you can work with the stability in lateral bending. So here are things that can help us with the primary stability, but most likely over time also help us to secure that it gets stronger and stronger. We mostly be working with above-knee amputees because they have a lot of problems with the sockets. But of course, osseointegration is a generic bone technology, so you can use it for thumb amputations. This is a below elbow amputation, and here you can see we can have sturdy tools, or we can have more human-like myoelectric prosthesis. If you have a very short stump, it's impossible to put any retention with the socket. And for instance, the shoulder joint, that is my favorite joint. It's the joint with the highest range of motion and very, very nice precision. With this technology, you can really create a new arm. Well, maybe. Another thing here is that, okay, biomechanically it's good, but what will it really mean for the patient? So this is from our collaboration with Department of Defense. And here you see it's a bilateral amputee putting on a socket prosthesis compared to putting on an osseointegrated prosthesis. It's still cumbersome, but the difference is dramatic. So those unfortunate that get this kind of devastating, traumatic things, we need to do everything we can to help them. So, isn't this fantastic? There is almost no arm, and still you can have this range of motion and do things. And then we said, oh, this is not good enough. This is not a real arm. This is a dead fish. So what can we do about that? How can we make that dead fish come alive and be a hand? Yeah, so that's something there's a lot of people in the world working on this. So it's a combination of osseointegration. You can see I underlined in red there. Something called targeted muscle reinnervation, muscle sensors, advanced algorithms, sensory feedback, peripheral nerves, etc. So I will now try to dig into that and go into what you might think is science fiction, and we see what you say in the end. So osseointegration, TMR, muscle electrodes, artificial intelligence, and nerve electrodes. This is Blake Leeper. He's an amputee. He's better than all of us on this planet, because he was not allowed to compete in the ordinary Olympics because he was too good. So Hugh Herr, that is the professor at MIT I work with, he's also a bilateral below-knee amputee. He sort of jokes, but not really jokes, and say, in the future, everyone would like to be an amputee because we will outcompete the rest. Do you think it's possible? You don't think it's possible, but I will show you it is. So first, we need to work with the brain. So the brain will think and feel that it's not a prosthesis, it's actually a part of the body. We attach it in the best possible way with osseointegration, but then we need to talk to the brain and have the brain talk to the prosthesis. So when the brain is signaling through the peripheral nerve system and the muscle system, we have electrodes there. We pick up those signals, we route them out through the implant system, we convert them to signals that we drive and have the hand do what the brain thinks. But we also need to have sensory feedback, because a hand without sensory feedback is again a dead fish. And we need to bring those signals back to the brain, so the brain thinks that the normal hand is there, and it feels. So what we have done that we have used this osseointegrated implant system and upgraded it with electrodes. So here is a contact, and there goes electrodes here. They go out through the bone. You see we have muscle electrodes, and here you have electrodes around nerves. And this is actually a real patient treated, and you see here again electrodes around nerves, muscles, and this is an X-ray, and this meshy thing here, that's all the wired connections. And here he is, the first patient in the world treated with this system, now more than 10 years ago, and he's showing first how challenging it is to control a prosthesis with electrodes on the outside on the skin, surface electrodes. Because the surface electrodes are picking up all the muscle activity inside the arm and cannot see the real important signals. So once he's starting to move the arm, he cannot control the grip. He can only control the grip when he's holding the arm still, which is not really a favorable situation. So here he's switching to the implanted electrode control system, and suddenly he has full control, not even with really thinking, intuitive control. So what about sensory feedback? That's actually very difficult. So this is one of the first attempts. So this is a hand that has pressure sensors in the fingers. There goes an electrical signal, and built into the arm we have a neurostimulator that pick up those signals and send a neurostimulatory signal into these nerve cuffs so he can start to feel again. Sensory feedback. Well, there is still a lot of work to do, but we also need to figure out, what is really important. So typically in the field of arm prosthesis, you talk about, okay, they should be able to play the piano. And I tend to respond, okay, I can't play the piano, so for me that's not important. So we need to figure out what is really important. And a big problem here is that a lot of research is being done on rats or monkeys or so. And if you ask them, how does it feel? You cannot get the real answer. So we really need to have pioneering patients to help us to understand what is needed. So here are a few more things to advance it even further. So if you lost the arm, the nerves are still there, but they don't have any target. So what if we take the nerves and suture them to other muscles? And that is what targeted muscle reinnervation is about. So here, the nerves that should go to the hand and the fingers, they have been sutured into the pectoralis muscle on the chest. This is technology developed by Todd Kuiken in Chicago. So this is Jesse, the first patient in the world to undergo that kind of surgery. And this is Todd Kuiken's hand. And on his chest, he's doing the same movement as Todd Kuiken's hand is asking him to do. And then you say, okay, I couldn't figure out that was, index finger movement. How can you use that? Artificial intelligence. It's super easy for artificial intelligence. This is easy pattern recognition to take those signals and drive a hand, an artificial hand. So this is the AMI concept developed at MIT and Brigham's. So what is this? Well, there's some muscle sliding back and forth. So how will this work? Can the brain really think that the leg is there if it's not there? This is an illustration from how the surgery is done. So this is a below-knee amputation. And you see there are muscles that are connected here. And you harvest these muscles when you do the amputation and put them and link them together. And why is that? Okay, so how do you flex your arm? Then everyone says, oh, you just fire your biceps. Okay, there is a small biceps in there. But that's wrong. So how do you control joint movement? Biceps and triceps. They need to work together. Otherwise, it will go like this. And that is exactly what they're trying to do here, to trick the brain to think that the ankle joint is still there because they're connecting these biceps or triceps for the ankle joint. And that is what's called the agonist-antagonist myoneural interface, the AMI. And here you can see how it should work schematically. So you have these constructs, and then they should talk to the external component, and it will be perceived as natural by the brain. Bit scary, but it's actually working. They have done some 50 cases now, and it's amazing that the patients feel that they can move their ankle joint that does not exist. So this AMI construct was originally developed to be used during amputation, when you had the intact muscles. Most amputees, there have been amputees, and there is no that opportunity because the muscles are not there. So can we create new muscles out of thin air? Yeah, that's the regenerative peripheral nerve interface. So you take a free piece of muscle without any vascularity, and you suture a nerve ending inside it, and you wrap it around, and you create a small burrito. And then it will be revascularized from the nerve. And if you do this on a big nerve, you split it into several fascicles. You put several of these RPNIs around it, and you take another nerve that is innervating the antagonist and do the same thing. And then you lock them together. Aren't we then creating a new AMI, a regenerative AMI that did not exist, but we had still the nerves there? So this is how it's working. You have the agonist and the antagonist, and then you put electrical activation, so you have a contraction on one side, then you have elongation on the other side, and that elongation should then be sensed by the brains as a natural elongation. And this is exactly what is happening, not only in rats where they started, but now in humans. Regenerative for those that don't have any muscles there, and native, they're a bit better and stronger because it's muscles that you keep during amputation. And to get this to work, I'm just checking how much time I have, and now we go into why MIT is important. So these are, control strategies to take those signals, drive an external prosthesis, get signals back, and trigger it so that the brain can feel that it's absolutely natural. And here, for instance, here you have the agonist-antagonist myoneural interface, and here you have a lot of control things. I will show you another thing. It's very new, so you cannot really see it, but here you see something called MCMIs. This is the problem with electrical stimulation of nerves is that it's very difficult to be absolutely natural because, electrical tingling of the nerve, we all had that when we hit here. That's not really a nice feeling, even if it can be downgraded. So how can we create absolutely natural sensory feedback? And there is only one way. to work with biology. So the idea here is to take a small piece of skin that is innervated, and you have all the mechanoreceptors. And then how can you trigger that? Well, you put this inside the body, and then you wrap a small piece of muscle around it, and when the muscle is contracting, it will press the mechanoreceptors, and they will send an absolutely natural, normal signal back to the brain. So this is work that's just started to be done in humans. I don't think there's any human that is already through it, so we don't know what the humans will say, but I think it's a very, very interesting path. So bringing all of this together, we see how important osseointegration is here. Because if we don't have this communication portal and good mechanical anchorage, there is no way we can do it. But this looks rather complex, and how can the patient really work with all of this? Well, this should be just plug and play for the patient. Just connect it and up and go. So here's a picture from the surgery. You see actually this is a muscle electrode just placed, and here you see all the wires that will be used for these different contacts. This is the only patient in the world that has a combination with this TMR, the target muscle reinnervation, and the small free muscle pieces called RPNI. This is not the patient that also has the AMI construct, so we can even add complexity to this. But here he is, and he's controlling the grip, the rotation of the wrist, and the flexion of the elbow at the same time independently. And that is intuitive control of 3 degrees of freedom, and that has never been shown before. This is above elbow amputation. No forearm muscles left that we control all finger movements, still individual finger control. Can't be done. Smart pattern recognition algorithms, artificial intelligence, and smart biological reconstructions. There are 2 patients in the world with this system on the transradial level, and here is the first one. And this is with another type of hand with sensory feedback. So those boxes, they have different stiffnesses, and the patient will try to grade them from softer to harder. And you see it's a speeded up video. A different type of sensory feedback, not touch, but stiffness. Okay. This is one of 2 patients with RPNI constructs above knee amputees treated at Brigham's. And this is the first time a patient with that amputation level can control, you see here's the foot, can control the foot by using thigh muscles. They don't control the foot. It's because there are RPNIs there and instrumented with electrodes. So science fiction is here today. And I hope you all would like to become amputees in the future, because then we can do much better. No, I hope you will not. There is still, it's a very, very tough thing to be an amputee. I worked with it for many, many years. I can't still realize what it's all about, but it's terrible. Anyway, we can help them. We should try hard. So, osseointegration is a very, very important part of what we can do in the future to drive this to heights we cannot see today. And of course, walking is a basic thing that we should try to bring everyone to do. But with the good mechanical anchorage and, you know, the proprioceptive feedback, or osseoperception, as PI like to call it, we can go further, we can do more advanced activities, because we get the feedback needed to control a non-existing limb in ways that no one thought was possible. And of course, by then adding osseointegration as a communication portal, and use that to talk to the brain indirectly, back and forth, we'll start to create something that, together with what I call the biological constructs, will really start to bring that dead fish that the prosthetic used to be, to become a new arm or a new leg. Thank you very much.